Hello. We'll start today with the functional anatomy of the hypothalamus and the pituitary gland. As we said in the last lecture, the pituitary gland is the master gland of the body. It is composed of two distinct parts, the anterior pituitary, which is the adrenal hypophysis, and this is the classical gland composed majorly of cells that secrete protein hormones. The posterior pituitary, which is the neurohypophysis, it's not really an organ, but it is acts as an extension of the hypothalamus and composed largely of axons of the hypothalamic neurons. In many mammals, there is intermediate lobe between the anterior and the posterior pituitary gland, which is called pars intermedia. The anterior pituitary collects its hormone from hypothalamus via the hypophyseal artery of the hypothalamus. Capillaries within the anterior pituitary, which carry hormones, is secreted by that gland, combine together into veins and drain into the systemic venous blood. Those veins also collect capillary blood from posterior pituitary gland. The anterior and the posterior pituitary gland secrete a battery of hormones, a lot of hormones, that collectively affect cells and affect all the physiological processes. Neuron within the hypothalamus, which is called neurosecretory neurons, secrete hormones that control secretion of hormones from the anterior pituitary. So the hypothalamic releasing and inhibiting hormones are carried directly to the anterior pituitary gland via, as we said before, via the hypothalamic hypophyseal portal system or portal veins. These hypothalamus hormones are influenced by emotions and metabolic state of the individual. Specific hypothalamic hormones bind to the receptors on specific anterior pituitary cells and modulating the release of hormone they produce. So they affect the release of hormones. So as an example, in cases of stress or cold, the hypothalamus is activated to secrete one type of hormone, which is TRH, thyroid releasing hormone. This thyroid releasing hormone released from the hypothalamus will act on special types of cell in the anterior pituitary gland, which is called thyrotropes. These thyrotropes have a receptor on their surface for the TRH. Once the thyrotropes is activated in the anterior pituitary gland, it starts secretes the TSH, which is the thyroid stimulating hormone. This thyroid stimulating hormone is carried by blood to reach to the target organ, which is the thyroid gland, ordering the thyroid gland to release the T3 and the T4 hormones, which is the main hormones of the thyroid gland. So you can see here as an example, generally the hypothalamus secrete releasing hormones, while the anterior pituitary release stimulating hormone. So from this figure, we can summarize all the types of hormone that is affected by the that lead to the activation of the pituitary gland to secrete its hormone. You can see that at the first level, the hypothalamus have the indirect control through the releasing of regulatory hormones. You have the corticotropin releasing hormone, the thyrotropin releasing hormone, you have the GHRH, growth hormone releasing hormone, the GHIH, growth hormone inhibiting hormone, you have the prolactin releasing factor, you have the prolactin inhibiting factor, inhibiting hormone, PIH, and you have, of course, gonadotropin releasing hormone. All these 7 releasing hormones affect only the anterior part or the anterior lobe of the pituitary gland. While you can see from the posterior lobe of the pituitary gland, mainly it is an extension, as you can see, it is the extension of the hypothalamic neurons. And as we said before, it have a direct hormone release through the sending axons of the sensory stimulation and osmoreceptor stimulation to the posterior lobe. Once the anterior lobe of the anterior pituitary gland got stimulated by the releasing hormone from the hypothalamus, it starts secreting its hormones. As an example, it starts to secrete ACTH, which will affect the adrenal gland and especially the adrenal cortex to release the glucocorticoid steroid hormone. As well, TSH will affect the thyroid gland to release the thyroid hormones. As well as a major gland, a major hormone, which is the growth hormone, which will affect one of its targets is the liver, leading to the secretion of some other hormones like somatomedians, which will affect bone, muscles, and other tissue. As well as the prolactin, which will affect the mammary glands. Another two hormones from the anterior pituitary, which is the FSH and LH, follicle stimulating hormone and the luteinizing hormone. Both of them will affect the gonads, either testes in males or ovaries in female, to release their hormones. In male, the testosterone and inhibin, and in female, majorly the progesterone and estrogen and inhibin as well. So these are the hormones of the anterior anterior lobe of the pituitary gland. While for the posterior pituitary gland, you can see that majorly there are two hormones, the ADH and the oxytocin. The ADH will affect the kidney to regulate the amount of water in our body, while the oxytocin affect majorly in female the uterine muscles and the mammary glands, while the oxytocin in male it affected the muscles in the ductus deferens, which actually the duct or vas deferens, it is the duct which carry the sperms, and as well will affect the prostate gland. Another hormone, which is MSH, melatonin stimulating hormone, it affects the melanocytes, and they have they don't know really the significance in healthy adult until now. So as a summary for that figure, it shows all the network of hormone regulation starting from the hypothalamus until reaching to the target organ. So again here you can see the spectrum or the battery of hormones secreted by the anterior pituitary gland and posterior pituitary gland. Actually, in the posterior pituitary gland, the hormones comes from the hypothalamus, and as we said, the posterior pituitary represent an extension of the hypothalamus. Again, the MSH, the melanocyte stimulating hormone, comes from the region between the anterior and posterior pituitary gland. For more elaboration on the axis of the secretion via the hypothalamic and the pituitary axis, you can see that in the hypothalamus, you have different neurons, which we call it neurosecretory cells. Each type of neuron responsible for secreting certain type of hormone, as we said before. This hormone pass into the pituitary, hypothalamic-pituitary portal vein in order to either amplify the signal represented by plus or diminish the signal represented by minus. So you can see the effect of each releasing hormone. As you can see in the anterior pituitary gland, for example, if the prolactin releasing hormone is secreted from the neurosecretory cells in the hypothalamus, it will potentiate the secretion of prolactin from special types of cell to affect the breasts. As well, this is inhibited by dopamine or PIH, prolactin inhibiting hormone, in a mode of regulation to regulate the amount of prolactin secreted into the systemic circulation. Same here for TSH, Same here for the other hormone and growth hormone. You need to understand that there is a specialization of these cells. So there is a special neurosecretory cells for each type of releasing hormone in the hypothalamus, and there is a special types of cells as well in the pituitary gland responsible for secretion of each type of hormone. At the ultimate target, once the target organ or cells are activated, it will perform its function. You can see the growth hormone here, for example, can act on different targets, but all of them will promote growth and control of a metabolism. Again, here we'll see the different levels and the integration through these axes of hypothalamic pituitary axis. It starts with the sensory inputs from the environment, which detect the need of the body for a certain hormone. This will affect the central nervous system to affect the neuroendocrine cells inside the hypothalamus. And this is called the first level, which is called the hypothalamic stimulation from the central nervous system. Soon after that, the hypothalamus, as we said before, it is either affect the posterior pituitary or affected through the release of releasing factors to the anterior pituitary. And this, the anterior pituitary or the posterior pituitary, represented the first targets. So you can see this pituitary stimulation from the hypothalamic trophic hormones. Soon after that, these endocrine cells exist in the anterior and posterior pituitary gland will be stimulated to release their own hormones. At the second target, this hormone released from either the anterior pituitary or the posterior pituitary will affect the second target, as we said before, specific for each hormone, until reaching to the final or the ultimate target. In case like the adrenal cortex and the thyroid gland, it will affect ultimate target. But in case like somatotropin and prolactin, there is only ultimate target, there is no step in between. You can see here as well that blood glucose level can be affected by the secretion of different types of hormone. So here you can see the overlap between the actions of the hormones. Again, an illustrative figure showing where is the portal system. You can see here the portal system and how the releasing hormone are conveyed from the hypothalamus to reach to the endocrine cells inside the anterior pituitary. And actually these are the same hormones we just have discussed in the hypothalamic hormones, in the anterior pituitary gland hormones. And after the release of these hormones, it goes to their ultimate target, and maybe some of them are non-endocrine targets like the breast. And you can see that there is a lot of integration that may affect a lot of many tissues. So how hormone how the hormone impacts on the target cell. So multiple hormone, as we said, like the growth hormone, can target some cells or tissues. Like for example, the growth hormone and thyroxine hormone all have receptors on many tissues. They stimulate generally the pathways for growth. The impact of hormones on target cells can be categorized based on the mechanism they do. So you have a synergism. Synergism, that's mean there is a multiple stimuli from different hormones that lead to increase in the effect of the total action of the target cell. So this synergism may be multiple stimuli leading to additive effect. For example, If it's a cortisone alone, for example, will make effect of +5, and the glucagon alone will make effect of +10, and the epinephrine, which is the adrenaline, will make an effect of +20. When all of them have act on one target organ or a tissue or a cell, they will have synergism, and the effect will be amplified to +35. So these, if you add them roughly, it will be +35. But actually, this is not happening. There is a synergism. That means the effect by a certain mechanism is amplified to reach +140. Okay, so there is an interaction of this hormone to have a higher effect when existing in together and affecting a certain organ. While antagonism, actually the famous example is the effect of glucagon, which opposes the effect of insulin. As you know, the insulin controls the blood sugar levels and lowers the blood sugar levels, while the glucagon increases blood sugar levels. So what's a permissivism? It's actually when a hormone cannot exert its full effect without the presence of another hormone. So actually these kind of hormones need a help from another hormone to give him the permission to work. So that's why it's called permissivism.
What does the number represent? And +5, +10?
The numbers represent like a rough estimation to make it clear the idea, okay?
Oh, okay. طيب.
So let's see an example for that on the targets, the impacts on target cells. So as you can see here, there is the effect, the line chart here shows the increase in time in hours versus the blood glucose level, okay? The blood glucose level. You can see here, these three hormones, when acts each one separately, of course, the glucagon will decrease the amount of sugar in blood. Cortisol generally have no effect, you can see, generally it's a stable level, have no effect on the blood glucose level. The epinephrine, of course, it increases the blood glucose level. So generally each of these three hormones, when working separately, each one will have its own effect on the blood glucose level. Once the glucagon is in action with epinephrine, you can see that the blood glucose level is affected dramatically. Another dramatic effect when the cortisone is present in action with the epinephrine and the glucagon, the blood glucose level is markedly increased. So this is a perfect example of synergism leading to the increase of blood glucose level. So let's here see an example of the permissiveness. You can see that the thyroid hormone will, this thyroid hormone can affect, when it reaches its target, you will not see any fatty acid released. That means the hormone cannot do its job. But while the epinephrine, when it reaches its target, it will release, as in the process of burning, of course, it will release a small amount of fatty acids. In the existence of epinephrine plus thyroid hormone, there is a large amount of fatty acids. So it's very clear that synergism is different than permissiveness. Why? Because in synergism, each hormone can work already. It's working already isolated or being, it have its own function. But here you can see that the thyroid hormone, there is no function at all. And it starts increasing the amount of the fatty acid release due to the presence of epinephrine. Okay? So let's see here.
دكتور باللي قبلها هي ليه synergism مش permissiveness؟
Which one? This one?
لا بالفيجر.
This one?
مش نحن قلنا إنه الثايرويد هرمون بالبيرميسيفنس، قلنا إنه الهرمون بدها هرمون تاني لتعمل الفول إفكت اللي إلها.
Yes.
فهون الثايرويد هرمون ما عملت شيء غير لأجل الأبينفرين.
تمام. Because it needs the presence of epinephrine to make its action. It needs the permission from the epinephrine to work. Okay.
Okay.
Okay. So let's see here the types of the neurosecretory cells. So we have said before in the last few slides that hypothalamus contain neurosecretory cells, okay, which release the releasing hormone. So here we'll discuss the type or show the types of these neurosecretory cells present in the hypothalamus. So we have first type is magnicellular neurons. Magni means it's a big one. This magnicellular neurons is responsible for affecting, controlling the posterior pituitary gland to release, of course, the two major hormones, the vasopressin and oxytocin, which will affect the kidney and the uterine walls and the mammals and the mammary glands. Okay, so this is the magni neurons. The other type is a paravisceral, varicella hypophysiotropic neuron. And you can see that these neurons will affect the anterior pituitary to release the tropic hormone, most of the hormone we already said. The third type of the hypothalamic neurosecretory neurons is the hypothalamic projection neurons. And this will go directly to the target tissue, like the sympathetic preganglionic neuron in the spinal cord. So generally these affect the sympathetic nervous system, which is one of the divisions of our nervous system. So mainly its target are neuronal targets, but the other targets here are either gland or a tissue, a specific tissue. Okay. So for this slide, it shows the different types of neurosecretory neurons inside the hypothalamus. So how there is a control of the hypothalamic pituitary target organ axis. So basically here this slide will summarize the types of hormone we will discuss in the in the next lectures. So you can see here the effect of the hypothalamus CRH affect positively the corticotrophs, which is a specific type in the anterior pituitary, to release the ACTH, which is a trophic hormone. So the anterior pituitary will release this ACTH to the blood. It will potentiate and have a positive feedback towards the target organs here, for example, the adrenal gland, to release the target hormone, which in this case is the cortisol, as an example, and its action is cell homeostasis and function. Of course, cortisol, when it's when its action is terminated, the cortisol itself will provide a negative feedback on two levels. It will negatively regulate the anterior pituitary, as well it will negatively regulate the CRH. In this second example, you can see that the TRH can positively affect as well the another type of the of cells, which is the thyrotrophs, to secrete the TSH. This thyroid stimulating hormone will affect the thyroid gland to release its target hormone, which is the T3 and T4. And this, of course, will affect the protein synthesis. As well, again, when the function of T3 or T4 is terminated, it will provide a negative feedback loop to affect the secretion on the level of the pituitary gland and the level of hypothalamus. You can see that there is another releasing hormone, which is the somatostatin. It will negatively regulate these axes. Another example you can see here, the gene LRH, once it released from the hypothalamus, it will affect the anterior pituitary gland to release LH and FSH. And we already discussed the function of LH and FSH. It will affect testes and ovaries, gonads, to promote ovulation and spermatogenesis. Once this function is terminated, there is as well a negative feedback loop which will work again on the both levels of the hormone secreted. For the GHRH, growth hormone releasing hormone, it will affect as well the release of the growth hormone from the anterior pituitary gland. So it will affect this hormone, which will have direct organ on the liver tissue to release the other important factors like the IGF-1 and the which will affect the formation of the cartilage in our as well. One of its target is the formation of chondrocytes, which is responsible for formation of a cartilage in our bones. The last one is the secretion of a dopamine, which will affect another specific type of cells in the anterior pituitary, which is called the lactotroph, which will release the prolactin to the general circulation, and it will affect the mammary glands for the process of lactation during breastfeeding. So we'll deal for each these axes in our course separately. So here is the model for the development of human anterior pituitary gland. As we said in the last slides, that the anterior pituitary or the pituitary gland have a different cells, and these cells are specific for a different function and actually for secreting for a specific function. So how this slide describes how these different types of cells have been produced. So in this diagram, it shows the model for development of the human anterior pituitary gland and the cell lineage determination by a cascade of transcription factors. So as you know, transcription factors, it is a group of proteins that sits on the DNA and initiate a transcription of a specific genes. So you can see here in our primordial cells, that's mean it is cells that is not differentiated to or still not committed to a certain fate, still it's a kind of stem cells. This under the effect of a number of growth factors and under the effect of another growth factor, OTX2, it is transformed to be, differentiated to be pituitary commitment. That means it's only will be involved in the pituitary function. Another effect of a lot of transcription factors, you can see, this lot transcription factor, it will lead to a proliferation and as well proliferation into lineage precursors. This proliferation upon the effect of another transcription factors, the different cell lineage of each type of a specific cell in the anterior pituitary will be differentiated. So you can see that the main role of these different transcription factors, which is either growth factor or transcription factor, will affect the fate of these cells to develop into specific type of cell in the anterior pituitary to be committed to secrete a specific type of hormone. You can see as well that is in a certain type like the corticotrophs, for example, it needs a combination between a different transcription factor. Some other cell types need even a combination, just a single signal coming from the transcription factor. Others requires 2 signal like the GATA2. So these slides, as we said, summarize how the different types of cells in the anterior pituitary gland are differentiated. And this majorly is affected by the type of the growth factor and the type of the transcription factor. And of course, all of these types of cells are originated from primordial cells through process of commitment towards the formation of a specific cell type. So here again, we'll summarize the model. So you can see that the different type of cells, gonadotrophs, thyrotrophs, lactotrophs, somatotrophs, corticotrophs. All of them, it shows here the time of appearance in the fetus. So they start their appearance in the fetus. Here you can see the type of hormone they secrete. Here you can see the nature of the protein. So all of them, you can see that the last three of them is purely a protein, but the first 3 hormones are glycoproteins. So there is a sugar moiety conjugated to the protein. Here you can see the stimulators for the release of that hormones. As well, you can see the inhibitors of these hormones. Again, you can see the target organs for this hormone, and you can see finally the trophic effect. So actually, what is the main function? So basically, this table is a very good table summarizing the hormones, the major hormones secreted from the anterior pituitary. And of course, here this is abbreviations for the table. So we will start now with the first hormone, which is the prolactin hormone. As you can see, this is the same figure we have just discussed on the mechanism of releasing of the prolactin. So the prolactin, or the lactogenic hormone, or the mammotropin, is a polypeptide hormone around the 200 or 199 amino acids. And until 1970, the identification of prolactin was elusive because it is structurally similar to growth hormone. So scientists until 1997 managed to separate between or identify the prolactin, as we said, because it's very similar in growth hormone. And as you know, it is secreted by a specific cell type in the anterior pituitary gland, which is called lactotrophs. These lactotrophs represent around 15 to 25% of the functional anterior pituitary gland. So it can reach up till like quarter of the type of cells in the anterior pituitary gland. Their number doesn't change with age. I mean the lactotrophs. But lactotrophs hyperplasia, this means increase in number, lactotrophs increase in number, occurs during pregnancy. Of course, it's obvious because it induces lactation. Okay, and this hyperplasia will be resolved in several months after delivery. Of course, the mother needs a lot of lactotrophs in order to secrete a lot of prolactin in order to induce milk in the breast for lactation. Most prolactin expressing cells arise from growth hormone producing cells. The prolactin is also synthesized and secreted by various immune cells, the brain as well, and the endometrium of the pregnant uterus. So here you can see another location for the secretion of the prolactin. Prolactin is synthesized as a prohormone. Anyway, generally, the hormones are first secreted as a prohormone. This following cleavage, the length of the mature hormone is between 194 to 199 amino acid, depending, of course, on the species. The hormone structure is stabilized by 3 disulfide bonds. So here we'll see how the prolactin in action. It has two mechanisms of action, either ligand dependent or ligand independent models. In the first one, the ligand dependent dimerization. You can see ligand dependent dimerization. So it means that the dimerization depends on the presence of the ligand. So you can see the prolactin hormone, once it reaches its target cell, it interacts with a monomeric receptor, which is called prolactin receptor, at the cell membrane of the target cell. Once there is an interaction of the prolactin on the extracellular domain of this monomeric receptor, this will induce, here you can see the interaction, one-to-one complex. This will induce a second receptor to be active, and hence the process of dimerization. So you can see the dimerization. That's why it's dependent on the ligand. So there will be no dimerization unless the prolactin is present. This dimerization will activate the intracellular domain, the region of the receptor which is inside the cytoplasm, which will lead to phosphorylation of important kinase, which is JAK2 kinase, that leads to the phosphorylation of the receptor itself. So again, the prolactin binds to one molecule. This induces dimerization. This dimerization will induce JAK2 activation and hence autophosphorylation, which will induce the phosphorylation of the receptor itself. The process of phosphorylation leads to the full activation of the receptor, which leads to the recruitment of another protein, which is STAT5A, to be phosphorylated. And at that time, now the signal has been transformed from outside to the inside until reaching STAT5A, which is now phosphorylated. So this is the action of the prolactin dependent on the ligand.
Doctor?
Yes? So this is the ligand-dependent dimerization. And as I said, the signal has been transformed from or transduced from outside to the inside, represented by the STAT5A, which is now phosphorylated. Because you can see that STAT5A in the cytoplasm is not phosphorylated. Once the receptor is activated by phosphorylation, this will recruit the STAT5A. And of course, the STAT5A will now have effect on other proteins inside the cell, the target cell. In the second mechanism, which is the ligand independent, you can see that prolactin reaches the receptor, and now the receptor is already dimerized. So that's why this dimerization is independent of the ligand. So the receptor already exists in the dimeric form on the cell membrane of the target cell in the absence of ligand. And in this case, it is still inactive. Once again, the prolactin binds now to the dimerized receptor. It will induce the same process of events until having the STAT5A being phosphorylated. So basically, these are the two mechanisms of the mode of action of prolactins on the surface, on the cell membrane of the target cell, and how the signal is transduced from outside to the inside.
دكتور يعني نحن الريسبتورز موجودين بتو فورم فينا بنلاقي هيك وبنلاقي هيك
The receptor is not in the two form. The receptors, it's only one form. It is only the mode of activation. So in first mode of activation, it needs the ligand for dimerization. But in the other mode of activation, it doesn't need the ligand for the dimerization. Anyway, for
كيف بصير
sorry,
كيف بصير الدايمرايزيشن بدون البي ار ال
There is another mechanism. There is another mechanism to control the dimerization.
Okay.
Okay. So let's see here the main functions of the prolactin. Of course, the milk production during pregnancy and lactation, reproductive and metabolic effects, mammary development, melanin synthesis. So the mammary gland development and the milk production and the reproduction, one of the major functions of the prolactin. So prolactin induces the growth of mammary gland. As well, it stimulates the nipples and the mammary gland during nursing to release more prolactin. The prolactin as well stimulates lactogenesis or milk production after giving birth. The prolactin plus cortisol plus insulin all together work synergically to stimulate gene transcription of the milk proteins. If there is a deletion or a mutation, the scientists have found that if there is a deletion or a mutation in the prolactin gene in heterozygous female mice, they show failure to lactate and will actually produce the half normal amount of prolactin. So to have the normal amount of prolactin, normally you need to have the 2 copies of the gene, not only 1 copy. In case of homozygous mice with inactive prolactin gene, they're actually incapable of secreting of prolactin, and they will be infertile. So you see here the connection between the prolactin and fertility. So that's mean if we want to expand the picture, this research shows if a female is not producing a prolactin, this will lead to be a infertile female, because she will have a defect in the process of ovulation, fertilization, and the development and the implantation. You can see that these all are responsible for the development of the embryo. So hence prolactin is very important as well. Prolactin also important in non-lactational aspects of reproduction. Yes, because as we just said, the absence of prolactin will affect ovulation, will affect fertilization, will affect the implantation. So these all are non-lactation effect. The prolactin is not only responsible for the production of milk during breastfeedings, but it is responsible for the development as well of the embryo and the fertility of the female. So it have another function which is non-lactation function. So what's the effect on the immune function? The prolactin receptor is widely expressed by immune cells. Some types of lymphocytes synthesize and secrete prolactin as well. Prolactin, as we said before, prolactin may act as an autocrine or paracrine modulator of immune activity. So if we look to the control of the prolactin secretion, we have a dopamine. The dopamine is the major prolactin inhibiting factor. The dopamine is secreted into the portal blood by the hypothalamic neurons, and it binds to receptors on the lactotrophs. So the dopamine binds on the lactotrophs. And of course, we have said that the lactotrophs is responsible for secreting of the prolactin from the anterior pituitary gland. So once this dopamine binds on the receptors of the lactotrophs, this will inhibit the synthesis and the secretion of prolactin. So drugs that interfere with dopamine secretion or receptor binding lead to enhanced secretion of prolactin. So if someone have an abnormality in dopamine level, she can take drugs that will inhibit the binding of dopamine to this receptor to enhance the secretion of prolactin. If a pituitary stalk is cut, what is a pituitary stalk? Actually, it is the region connecting between the hypothalamus and the pituitary gland. So if the pituitary stalk is cut, the prolactin secretion increase, while secretion of all other pituitary hormones, will fall dramatically due to the loss of the hypothalamic releasing hormones. So here you can see, so here we'll discuss the mode of control of the prolactin secretion. What are the factors which promote its secretion, and the factor which down-regulate the secretion of prolactin? As you can see from the figure, here is the pituitary gland, here is the anterior pituitary gland, here is the pituitary, the stalk that connects the pituitary gland to the brain. And you can see that majorly we are talking now about the secretion, the regulation of secretion of prolactin from the anterior pituitary gland. So you can affect the major effectors of these are the TRH, which potentiate the secretion of prolactin, and the dopamine, which inhibit the secretion of a prolactin. You can see as well important thing here we'll discuss later in the next few slides, that the pituitary glands sits under the optic chiasma. And as you know from previous lectures, the optic chiasma is the region of intersection between the two optic nerves of the eyes. So you can see as well that estrogen have a positive effect on the secretion of the prolactin. And the estrogen is affected as well by physiological causes, which is, of course, one of the major physiological causes is a pregnancy, which promote the secretion of estrogen, which will potentiate the secretion of prolactin. Some of the factors that inhibit the secretion of dopamine, so once inhibited the dopamine, that would lead to increase in the secretion of prolactin. The inhibiting of dopamine release leading to the reduced inhibition and the increase, of course, prolactin action, the prolactin production. These negative regulator can be medication. So there is a lot of medications that lead to inhibition of the dopamine. One of the famous modification, one of the famous medications is the anti-ulcer agents or the H2 blockers, the one which we take it for anti-acid. And as well, the antidepressant will decrease the amount of dopamine in our system. This will lead to increase in prolactin. Another negative regulators of dopamine is a neurogenic effect, like chest wall injury or breast stimulation or breastfeeding. This via autonomous nervous system will decrease the release of dopamine and hence increase the secretion of prolactin. Another effect which will potentiate the TRH, which in turn will secrete more prolactin, is the hypothalamic prolactin stimulation through primary hypothyroidism or adrenal insufficiency. The rest of this figure shows that there is another factor which potentiate the prolactin, like some of the syndromes, like the polycystic ovarian syndromes or pituitary tumors. So if someone have a pituitary tumor, like the adenomas or hypothalamic stalk interruption, as we said before, if you cut the hypothalamic stalk, it will increase the production of prolactin. And the hypophysis, like inflammation, all these cases, you can see the difference in figure, will lead at the end on the increase of the secretion of prolactin. As well, if you have reduced prolactin elimination, that's mean, because of course hormones need to be degraded and excreted once the function are terminated. So there is, if there is a reduced prolactin elimination from the circulation, that will lead to, due to either a renal failure or a hepatic insufficiency, that means it's unable to do its job, this will keep a high level of prolactin in the blood. Also, you have some abnormal molecules that can bind to the receptor of the prolactin and initiate a cascade of reaction, which will mimic the effect of prolactin and hence will potentiate the effect of prolactin. In these cases, you have a case like macroprolactinemia, or this macroprolactinemia. It means that the binding of a molecule, abnormal molecules, binding to the receptor of the prolactin will have the same effect as if the prolactin in the circulation. As well, if there is a lot of receptor have been amplified. So in this case, you have the individual have a genetic effect which will expose the receptor of prolactin in amplified, and you can see multiple copies of the receptor. So as we said, this figure explains the mode of regulation of prolactin and how the different factors affect the action of prolactin and the level of the prolactin in the blood and the regulation of the prolactin. Keep in mind that it is not necessarily that all these factors occur at the same time. But here in this slide, we are collecting the major causes of prolactin regulation. You can see that there is another causes which is still unclear and can affect as well the prolactin levels in the blood. So let's talk about an example of a pituitary tumor and how it affected the secretion of prolactin. Okay. So let's see an example of how pituitary tumors can affect the secretion of prolactin. The excessive secretion of prolactin is called hyperprolactinemia. Hyperprolactinemia. This condition has numerous causes, as expected here, including the prolactin secreting tumors, and the therapy with certain can be done by certain drugs. So here you can see, in a woman, the hyperprolactinemia causes amenorrhea, which actually the lack of the menstrual cycle, and it can cause as well galactorrhea, which is actually the excessive or spontaneous secretion of milk. So the female in these cases or in this condition can have milk dripping from the breast spontaneously. In men, the hyperprolactinemia causes hypogonadism. So what's meaning of hypogonadism? It's mean it will lead to decrease in the sex drive, is not willing to do sex, decrease in the sperm production and impotence. Such men often show breast enlargement, gynecomastia, but very rarely produce milk. So you can see here, the figure shows that's an MRI, magnetic resonance imaging. That's an MRI showing the normal view of the region of the brain which contains the pituitary gland. And here the other MRI shows an enlarged adenoma, which is a type of tumor in the pituitary gland. So you can notice the difference here. So here is the region of the pituitary gland here between these two carotid arteries. These are the carotid arteries, which are the arteries supplying the brain with the blood. So you can see that here the pituitary gland sits between the carotid arteries. Above it straight, there is the optic chiasma. So in the figure here is the two black dots, and here is the pituitary gland. You can see it. And this one is the optic chiasma. But here in the case of the tumor, you can see how the tumor is enlarged. So you can notice the major difference between both of these. So what do you think? When this tumor have been enlarged, it will affect straight away. It will affect the structure which is above the pituitary gland, which is obviously the optic chiasma. So in this case, this adenoma, we call it macroadenoma, big, macro, big. And this will lead to elevating the optic chiasma. So the increase in size of the adenoma will push upward on the optic chiasma. And this will interrupt the signal for the visual signal from reaching the brain. Okay? So you can see here how the effect of the prolactin on visual effect. So in normal vision, as you can see, in normal vision, the right eye send their signals to the left region of the brain, while the left eye send the signal on the right region. And you can see the pituitary gland lies just beneath the optic chiasma. Okay, so generally the region of the visual region is divided into two fields. The inner one toward the middle is called the nasal visual field, since it's near the nose, while the other one it means the temporal visual field. Okay, so in case there is a tumor, you can see the tumor, it will push on the blue nerves, the nerves represented in blue, which is responsible for the temporal field region. So the tumor will pushes over the chiasma, will block the signal. So individual having these cases will lose at all the temporal vision, will lose the temporal field vision. You can see. Okay. So again, in the normal vision, usually the nasal visual field with the temporal visual field of the two eye, by processing in the brain as a normal vision, this will lead fusion of the signal coming from the eye to see one complete picture. But in case in tumor, you have a lesion here, so it will affect the optic chiasma, as I said. This will lead to bitemporal hemorrhagic defects. So actually, a patient in these cases will have half of their each eye black. They don't see it, okay? Either they see it as this case or this case. It's all examples of fusion failure, or this case or this case. And all these cases depend on this, or either in the worst case, they see half of the eye is black. In all cases, this depend on the size of the tumor, which is pushing on the optic chiasma. And this explains how any abnormality in the size of the pituitary gland, as an example here, the macroadenoma, will lead to its major effect on vision.